organisers for the opportunity to come and talk. I was actually invited last year, but I couldn't make it, so I'm very grateful to have the second chance. I'm going to give you a perspective from the diagnostics organisation. So obviously you've heard from pharma and from an NHS service perspective. So I'm going to try to pull together some of those strands and really give you a little bit of history as well around the evolution of stratified medicine. If I can just learn how to drive this thing. Okay, so having spent a long time in the pharmaceutical industry myself, it's interesting over the last 10 years on how the balance has changed between the diagnostics and the pharmaceutical industry. And this is a statement that came from the FDA this year, actually, from Janet Woodcock, that the upcoming years may well be known as the age of diagnostics. And I think certainly when I started in the pharmaceutical industry, diagnostics was perceived as a bit of a country cousin. And I think that balance has changed significantly, and now diagnostics in many respects is the rate limiting step in a lot of pharmaceutical development work. But I really want to step back a little bit further than that and sort of talk about the evolution of how diagnostic technologies have improved over the years and how personalised medicine actually saves lives. So if you think about 100 years ago, if somebody went into a physician's office, then they could probably diagnose that this patient had something wrong with their blood. Over a period of time, diagnostics have improved. Whereas about 80 years ago, you could differentiate between different types of disease, whether you had leukemia or lymphoma. That evolved into subclassification into different types of leukemia. Whereas today, we have 38 different types of leukemia, 51 types of lymphoma, all of which are really determined by the availability of more sophisticated diagnostic technologies. And the more important thing is that the 5-year survival of these patients has increased from pretty much 0 to about 70%. So I think you can correlate, there's some significant correlation between the improved survival and the improvement in availability of diagnostic technologies, and obviously knowing what the technology, what the disease pathology is, then you can use the appropriate drugs, and then the drugs have improved that. So there's obviously had a clear benefit. But the statements out there, the stratified medicine and so-called theranostics are a new concept. I would challenge that actually, because if you look historically in some therapeutic areas, you'll find that stratified medicine's been there for some time. This is some data for HIV. What we've got here is 2 different patients on different drug therapies. So one of these patients is an HIV patient who has normal HIV. And as you can see, this is the blue line. So as this patient is treated with therapy, the viral load decreases down to a baseline level. Very good response to therapy. Then you have another patient who's a patient in red. Now this patient is a patient whose HIV has mutations which are associated with drug resistance, and you can see that using the initial drug combination that this patient was on, there was very little improvement in the viral load until the genotypic sequence analysis was carried out after about 10 weeks, where it was realised that this patient did indeed have resistance mutations, and that we should change the therapy to drugs that actually worked in this patient. And as you can see, when they changed the therapy to a different combination in the patient in red, then you got a much better drug response. So that's a real example of stratified medicine in action, if you like. And this is just an illustration of the complexity of this that's been around for certainly 10 or 20 years within the HIV field. So this is just a schematic of different HIV protease inhibitors and the resistance mutations that are genotypically recognized in the HIV protease gene, which are associated with resistance against specific protease inhibitors. And you can see the patterns are very different. And these patterns have all been interpreted into quite complex algorithms with neural networks. These patterns can be used. These patterns can be used with algorithms, for example, with other gene mutations in other HIV target genes, so that you can get now a very complex and very efficient algorithm in place, which will give you a very clear determination for an HIV patient as to which genes, which drugs should be used in those individual patients at each particular time. But obviously, as the virus mutates while patients are on therapy, then you have to continually monitor for changes in these mutational patterns so that you can adjust the therapeutics as the patient progresses through the clinic. Okay. Now, the other important thing to recognise within the HIV field is that there's an increasing prevalence of these resistant mutations because the antiretroviral drugs have been out there for so long that Actually, it's quite difficult to find normal HIV in the clinic these days, that the majority of circulating HIV strains are already resistant to a large number of drugs. So, for example, this is some data that's actually quite old now, that in 1999, 12,000 HIV patients in the US, a quarter of those patients had viral resistance to 3 major classes of HIV drugs. What this indicates that obviously you need careful monitoring for these individual patients. So this is classical personalized medicine. You're monitoring the patients as they're going along. That you need careful monitoring to implement it during the therapy of these patients, so that when new mutational patterns appear, you can change the drug accordingly. Otherwise, you're wasting money in terms of using expensive drugs in patients who are not going to respond, and the patient is obviously not going to benefit from drugs to which their virus is already resistant. The other important characterization is that preliminary characterization of all individual HIV patients now must be implemented before therapy is initiated. So this is in the guidelines. So this is a real step of an indication of a requirement from the regulatory authorities that for an HIV patient who comes into, who has not previously been treated, before any drug is used, then these patients have to be genotyped before the therapy is initiated. So I think that's an important concept that we need to keep in mind. And actually, regardless of what the pharmaceutical companies, what the patients are thinking, the regulatory authorities are now requiring this to happen, that these tests must be done before therapy is initiated. And they're also requiring that these type of biomarker studies are incorporated into clinical trials, so that any submission of a new drug has to be associated with as much information regarding the biomarkers within this area as possible. So, I think there's a lot to be learned. So we've had at least 20 years of experience in terms of personalized medicine in the HIV field, which can be correlated into other therapeutic areas. And I think what's happened recently, particularly in oncology, is that there's been a much bigger sort of exposure to cancer being a much more prevalent disease globally, to the importance of stratified medicine in that area, and that's really brought things to the fore. But I would also say that within HIV, what people perhaps forget is that the first HIV drug resistance test was cleared by the FDA in 2001, and this was actually the first IVD pharmacogenomic test that ever received FDA clearance. So the precedents are all out there, and I think it's incumbent on anybody who's working in the other therapeutic area to recognise what happened, what is happening in that area, and to use that information going forward within other viral diseases, but also in other therapeutic areas. Right, so switching gears away from virology for a little bit, and this is some therapy efficacy data from a number of different therapeutic areas. And as you can see, that in some therapeutic areas, for example in analgesics, the therapy efficacy rate is actually quite good. So if you go right down to the bottom, particularly in oncology, you get a very poor therapy efficacy rate. So from the pharmaceutical industry's perspective, that's really, well, from everybody's perspective, that's really not a very acceptable position. So, thinking about oncology and what's happened in the last few years, so I want to just briefly take you through some of the basic biology here. So a lot of the drugs that have come through, which are relevant to personalized medicine as we know it these days, are related to drugs that inhibit the epidermal growth factor receptor pathway. And there are a number of drugs in this pathway, which I want to talk about, particularly Iressa, which is a monoclonal antibody, which targets the EGFR receptor, and 2 other drugs, Vectibix and Erbitux, which are also humanized monoclonal antibodies which target particular elements of the EGFR pathway and relate to colorectal cancer. And what I do want to keep in mind is that these pathways are quite complex, but actually you can understand that any drug that's affecting the EGFR pathway is likely to have interactions with the individual components of that pathway. So, for example, if there's a drug who's manifesting its efficacy through the left-hand pathway, if you like, then biomarkers associated with RAS, RAF, MEK, and MAPK are likely to have some relevance to the way that drug operates. So just keep that in mind as we go forward. So I'm now going to show some real data. So this is the data that came out for Iressa. So this is AstraZeneca's non-small cell lung cancer antibody. What we've got here is an analysis from clinical trial. I think when AstraZeneca were developing this drug, they realized that they were doing, as they were doing global clinical studies, that there was a big difference in the response of patients in the Far East to patients in the rest of the world, which sort of sounded some alarm bells as if there might be some genetic component in here as to what the difference was between these cohorts and the responses in the individual cohorts. What we've got here, so in the middle of the graph in yellow, we've got progression-free survival of patients with non-small cell lung cancer who were treated with standard therapy, which is carboplatin and paclitaxel. And these are subdivided into patients who had normal EGFR and patients who had EGFR which was essentially full of mutations. And then you see there's actually very little in terms of clinical difference between those two strands. But when you look at the two green lines, so the green lines are the response to patients who were put on Iressa or gefitinib as it was known in those days. And you can see that patients who had mutations in the EGFR gene, in effect, had a much greater benefit from this drug in terms of progression-free survival compared to those patients who had normal. I think the mean, so you're coming down to probability of progression-free survival about 5 to 6 months for patients with normal EGFR compared to about 20 months with patients who had mutations in EGFR. So that was the first indication that this drug only worked in patients who had mutations in the EGFR gene. And that was obviously correlated in clinical trials, and that was incorporated into the label. So analysis of EGFR status by whatever means, whether it's by direct sequencing or by more sophisticated technologies, is required before this drug is prescribed. About the same time, Amgen in particular were working on on a drug for colorectal cancer, which targeted the EGFR pathway. And they submitted some data in 2007 to EMEA, who basically threw the drug back at them and said that the efficacy data is not good enough for this drug. We're not going to license it. So Amgen went back and they recharacterized every patient in the clinical trial. And in this particular study, they correlated they analyzed the status of a gene called KRAS. So what you've got here is again progression-free survival for patients who had normal KRAS as opposed to those patients who had mutants in KRAS. So this is slightly different from the AstraZeneca situation, because in this situation you can see that progression-free survival is associated with having a normal gene. So the blue line shows progression-free survival, the yellow line shows patients who had a defined mutation in the KRAS. So again, when Amgen analysed this data, they retrospectively analysed the cohort data, went back to the regulatory authorities in Europe and said, here's the data showing patient stratification around the KRAS gene, and you can clearly see that if you use this drug in patients who have normal KRAS, then you do get therapeutic benefit. So they did get approval. Okay, so as I say, unfortunately, the KRAS data doesn't answer all the questions. So there have been a lot of further work done on analysing the rest of the pathway to understand what's going on in those patients who apparently have normal KRAS but don't respond to the therapy. And what you can see for both Erbitux, which is the equivalent drug from Merck's Serono, and compared with Vectibix, that you can see that both these drugs are only effective in about 10 to 20% of resistant metastatic colorectal cancer. So 60% of colorectal cancers have normal KRAS, about 40% have mutants, so obviously they correlate with a poor response to therapy. 14% of those patients have a mutation in a different gene in that pathway, which is the BRAF gene, which is the next gene down in the pathway. But about 52% of those patients who apparently ought to respond to therapy don't. So that is probably associated with other biomarkers that we don't yet know about. But the take-home message is that as time goes on, we're having to accumulate more and more biomarker information to understand those patients, and it's going to come to a point where we have more and more of this information, so you're going to have much more complex biomarker studies which are required to be carried out, and data that needs to be incorporated so that you can really stratify in great detail those patients who are likely to respond to these drugs. Okay, so moving on to a slightly different perspective, this is another indication. This is metastatic melanoma, and this comes back to something we was talking about earlier, in which drugs are now being tailored to specific mutational patterns within individual genes. So this is a really quite exciting new drug, which was actually developed targeted for metastatic melanoma, and it was targeted against the BRAF protein, which we talked about earlier, as a mutational marker for colorectal cancer. So Roche developed, Roche and Plexicon worked together to develop this drug, which is a drug which actually targets a specific BRAF mutation, which is called the V600E mutation. And what you can see here, again, is a progression-free survival chart for patients comparing standard of care, which is dacarbazine, compared to patients who have vemurafenib. So as you can see, there is a significant benefit to these patients, and really, it's a huge step forward in terms of the treatment of metastatic melanoma. But this is a situation, this drug was targeted, this drug doesn't work against normal BRAF patients. So if you test a melanoma patient and they've got normal BRAF, then this drug will really not do anything very much. But in patients who have the specific V600E mutation, then you can see there's a considerable benefit. And this can be manifest itself very quickly. So what you've got here is a whole body scan of a patient with metastatic melanoma. So the figure on the left is the patient before therapy, and the black nodules are metastatic melanoma disease. The figure on the right is the same patient within 2 weeks. So this drug has a really, really dramatic effect. So it's a very successful drug, and it really shows the stratified medicine in action, if you like. That if you get the right mutation, you get the right patient, you get the right drug that's targeting that mutation, you can see huge clinical benefit very quickly. Okay, so what does this effect have on the pharmaceutical industry? So obviously we've got indications here that the regulatory authorities are demanding biomarker information. You've got clear indications that if you use the right drugs in the right patients, you get a much more efficient process. But this is some data, I think it's similar to some data that we saw earlier this morning, which is the R&D productivity of new medical entities per $1 billion of R&D spend. So despite the fact we have huge improvements in terms of combinatorial chemistry and the biology and the targeting and understanding the disease, even so, you can see that there's been a gradual erosion, quite a dramatic erosion in terms of the efficiency of developing new drugs. But one of the interesting figures within that says that actually, if you do retrospective analysis, you'll find that about 80% of all candidate drugs currently fail in clinical development might actually succeed if they were tested on the right patients. So I think that says it all. I mean, that's a huge step forward, and there's a whole industry actually being arising from retrospective analysis of failed drugs, going back and testing those drugs in patients who probably have the right biomarker character, biomarker profile to be able to respond to those drugs. So just by simple introduction of stratified medicine, I think it's going to have huge benefits to the pharmaceutical industry, and it obviously is, regardless of whether the pharmaceutical industry accepts that they have a problem. I'm pretty sure that everybody does now. The regulatory authorities are actually demanding this sort of information to be included in the clinical trials, and it should be beneficial for everybody. So, from the diagnostics perspective, the key thing here is, and I think to some extent the pharmaceutical industry has been a little bit panicked by this, that it's really important that when a drug gets licensed, the appropriate diagnostic test is available. And when I say available, it must be a diagnostic test that is fully validated, has regulatory approval, and is available in the parts of the world where that drug is going to be used. So it's very important to make sure that the drug discovery pipeline, the drug discovery timeline parallels the development of the diagnostic. And although this is, so that this slide shows you the stages in which a drug is developed and a comparable pathway for the development of the diagnostic, temporarily these are different. So we could expect that probably from preclinical through to launch is probably in the order of 10 to 15 years for a pharmaceutical. For the diagnostic, it's really not that long. I mean, there are examples where you can go from concept through to regulatory approved product within 6 months. So it's a very condensed timeline, if you like. But it's still important that there's a really close interface between the pharmaceutical companies and the diagnostic companies to make sure that when the drug is launched, there is a regulatory approved product which is out there and available. And this has been sort of exemplified quite recently. So now within the US, so the FDA are looking at parallel tracking of the approval process for the drug and for the diagnostic. Going back, this is the metastatic melanoma drug. So this was approved back in August. And interestingly, in this situation, Roche, who developed the therapeutic for the melanoma, also developed the mutational test through their diagnostic company. So these two processes happen in parallel. So the FDA approved both the diagnostic and the therapeutic at the same time. There was another example around the same time. This is for a new drug which was developed by Pfizer called crizotinib, which is a drug for non-small cell lung cancer, which uses a different biomarker linked to a rearrangement of the ALK gene. In this situation, Pfizer developed the drug, but Abbott was the developer of the diagnostic. So this companion diagnostic program was happening in parallel within Abbott as the therapeutic was being developed by Pfizer. And so again, there was a really close interface between those two programs to make sure that everything was done in parallel, and that the drug and the diagnostic were reviewed by the FDA and came out the other side together. But another thing that's happening really, which I think is going to completely transform the diagnostics industry, is the emergence of next-generation sequencing. We've heard a lot about sequencing this morning and the cost that's associated with next-generation sequencing. I can tell you within the US at the moment, for 3 specific mutational marker assays like KRAS, which come in around $600 each, you can get a whole genome sequence done. So it doesn't make a lot of sense to be targeting those specific genetic tests in the long run. But I think it's going to become a position where you can generate whole genome sequence for these patients at an economical cost. Whether you use all that information or whether you just target and pick out specific biomarker information is another point. But we as a company have set up a clinical genomics center in the US where we're associated with an oncology translational drug discovery unit. And we are now doing whole genome sequencing on every patient that comes through clinical trials, with a view to actually whether we use that data concurrently with the development of the drug, or we just store it retrospectively for analysis of response to therapy or clinical outcome. So it's a big area, and I think the drug approval agencies are encouraging this greater use of bioinformatics and looking at genotypic variation patterns, for example, to associate with response to therapy. And I think the emergence of regulatory approved next-generation sequencing is going to be a huge step forward within the industry. So one of the other things that the pharmaceutical industry needs to keep in mind, as I said, I touched on earlier, is that it's all very well to be working with a companion diagnostic so that diagnostic comes out the other end at the same time as the drug is licensed. But you also need to be able to physically do that test. And Pfizer basically demonstrated the way that this shouldn't be done with a drug called Maraviroc, which is an HIV drug which inhibits the attachment of HIV to the cells. Now they commissioned a lab to develop a companion diagnostic test. Unfortunately, the lab that developed the companion diagnostic test was not prepared to basically distribute that test to any other labs. So Pfizer got into a situation that they wanted to sell this drug globally, but the only lab in the world that could do the test was based in San Francisco. So that created huge issues for Pfizer because patients from all over the world who they wanted to use their drug had their samples logistically transported to San Francisco, and all the infrastructure that was required to develop that was a major issue. So that's something that is a model that the pharmaceutical industry really needs to keep in mind when you're working with a diagnostic company who is developing a new biomarker assay. You need to make sure that that company is able to get that test manufactured, that it is able to get through whatever clinical validation studies are required, that it's able to get it through regulatory approval, and to get it distributed throughout all areas of the world where it's required. So the other thing I wanted to touch on was the big differences between regulatory approval in Europe and regulatory approval in North America, which is a very different scenario. Now, in Europe, from a diagnostic perspective, To get regulatory approval in Europe, or a so-called CE mark for diagnostic test, there are the straightforward characteristics that you have to be able to demonstrate that the test does what it says on the label, that the sensitivity, the specificity, the robustness is adequate. There is no requirement for clinical validation for regulatory approval in Europe. So from a pharmaceutical perspective, it's actually a shortcut to get your drug licensed, just because there's a much easier process to get a regulatory approved test in place in Europe than it is in North America. So you can imagine that a lot of drug companies are factoring that in terms of the phased release of their drugs, to get it out in Europe, because it's going to take a lot longer to get full regulatory approval in the US. Now, there is a way that you can get it done in the US, which is a modification of the system in Europe. Now, so there are basically two different bodies of regulatory approval within the US for a diagnostic. There is a so-called laboratory developed test, which is equivalent to the sort of homebrew assays which a lot of labs in Europe run. But then there is full regulatory approval, which has different levels of sophistication depending on the number of clinical validation studies that are required and so on. So this is a definition of what a laboratory developed test or a homebrew assay is in the US. So it's an assay or a kit which is developed by the laboratory or the institution itself. It's regulated by a different body than the FDA. It's regulated by a body called CLIA. There's no official requirements for the standard sorts of things that you do during the diagnostic development process. There's no requirement for design control, no requirement for clinical trials. You don't have to have on-site manufacturing. You don't have to have quality control or review, and it doesn't need to have a recognised regulatory body to authorise the use of that test. There's no requirement for clinical validation, but it can only be used, that test can only be used in the laboratory where it was developed. Okay, so these are the sorts of things that test has to conform to. So it's pretty similar to what I talked about in Europe, that obviously that test needs to be able to show reproducible levels of accuracy, precision, sensitivity, and so on. Now, as I said, so actually within the US, there's an ongoing debate now about the levelness of the playing field, if you like, because obviously for an IVD manufacturer who's going down the route of taking their tests through the FDA, this can take several years. It can take many millions of dollars to get that test fully developed and fully validated and get full FDA approval. Whereas the CLIA certified laboratory, which can be enforced by the FDA, they tend not to regulate the development of this test. They leave that to this other body called CLIA. So it's a much simpler process. So from a pharmaceutical company who's developing a stratified medicine tool, you have these two options: that you can go down the IVD route, which is going to take years, which you may have to pay for, which the diagnostic company may have to pay for, or if you can identify the right sorts of labs who have the quality and the ability to develop those labs without going through that full regulatory approval process, you can sort of shortcut the development of the diagnostic test. And I think the consensus model is that most pharmaceutical companies are going down both routes. That they're taking the shortcut route through the CLIA lab to get this thing on the market as quickly as they can, so that they can use the drug, they can get sales of the drug. But actually at the same time they're going down the full IVD route as well. So what this has resulted in is the so-called emergence of vertically integrated diagnostics companies. So these are diagnostic companies who are essentially clinical service labs, but also have diagnostic capabilities. So these are usually independent labs. They're platform agnostic, which means they can do the test on pretty much any platform that's available. They have the capability to develop the test, and that's normally at the moment, I would say, either a protein-based biomarker, which is a simple protein-based assay, which could be an ELISA test or even a histochemistry assay, but also nucleic acid-based tests, whether it's using next-generation sequencing or real-time technologies. But they also have accredited laboratories in key global locations, so that where you can fully optimize and validate the assay, where you can actually run the clinical trial, so they can work with a pharmaceutical company during the clinical trial to validate the concept, to make sure that that biomarker does have relevance to this disease, and to make sure that the quality of the biomarker assay that comes out the other side is happening, is fully validated and fully compliant with the requirements of the clinical trial. And then to be able to run that assay as an LDT at the same time as turning that diagnostic test into a fully regulatory approved clinical test. The other thing that they need to have is have a full regulatory manufacturing capability, Because the last thing you want to do from a pharmaceutical perspective is to be working with a diagnostic company who's running your clinical trials, developing the assay, get to the end of the study, and then they have to pass the process on to into another body, to another manufacturer. It's much simpler to have it all done in one place, so that you can take the development program and you take that diagnostic marker right through manufacturing and regulatory approval within the same organisation. They also, of course, ideally would have the capability to take the product through full regulatory approval for 510K or PMA purposes as required. And as I mentioned a few times, they must have the global sales and marketing distribution network to cover all geographies, because you don't want to get into a situation where you have your test available in very limited regions, and where you have to put in very complex infrastructures to cover the transport and the transmission of data and all the rest of it. So really, to conclude, I think the take-home message is that the pharmaceutical industry and the diagnostics industry are coming very, very close together now. That synergy is extremely important to maximise the efficiency of stratified medicine. I mean, it's important to both parts of the industry. It's also critical that all the parties adopt a far more stringent approach to protection of intellectual property. And that's a major issue in parts of Europe, particularly in the UK. There's no incentive for a diagnostics company to work with a pharmaceutical company to develop a new diagnostic assay, which then some laboratory in the NHS is going to say, I'm not going to use that test, I'm going to develop my own version of that. And I'm going to completely disregard the intellectual property and develop that as a homebrew assay, because that is not in the interest of anybody, either the patient or the pharmaceutical company, because you get unreliable data, obviously from the diagnostics company's perspective, because they have no protection over their product. So there are a lot of lessons to be learned from the HIV area and latterly from other virology areas where personalised medicine has been in place for many years. I think the other thing is that diagnostic companies or pharmaceutical companies are perhaps thinking that diagnostic companies are needing to change their business model as they develop new companion diagnostics. And that's not the case, actually, because a companion diagnostic is just the same as any other diagnostic. The difference is that that companion diagnostic is going to be physically linked to the sale of that drug. So from a diagnostics company's perspective, it's actually quite beneficial to have that diagnostic linked to a particular drug, because it means that you have a regulatory approved requirement to use that diagnostic. So it's an enforced market, if you like, for the use of that diagnostic. And ultimately, there's no question that there are drivers here from all aspects, from healthcare providers, from government agencies, from pharma and diagnostics companies, and most importantly the patients, because as you can see that having this stratified medicine approach means that the right patient gets the right drugs at the right time, at the right dose, and all the rest of it, and it really maximises the value that's going to come out from the use of stratified medicine going forward. Thank you very much.
Any questions?
Hi, so my name is Ian Tote from the James Sutton Institute. I'm a microbiologist and plant scientist, and one of my interests is genomics and genome sequencing. Genome sequencing of bacteria, which are probably 5 million base pairs, tiny things compared to humans. But we also, at the Institute, we have people that undertake genome sequencing on things like barley, which is 5,000 million base pairs. It's huge, and it takes getting the sequence is relatively straightforward. Analysing the data and putting it all together is a very, very big task. And storing it takes many, many terabytes of storage. In fact, it has been easier to put it onto a hard disk and jump on an aeroplane to store it where it's stored in Ireland than it is to try and get it through the internet, the time it takes. And we're talking about a single genome. So although it's very straightforward now to sequence human genomes, in reality, how easy is it to look at 100, 1,000, 10,000 genomes and expect to store them and be able to gather the information, to back that information up, and to exchange the information over the internet?
Yeah, no, I think it's a good point, and actually it's something that's come up earlier. So, as I said, so we've recently we've launched our clinical genomics centre in the US. So, as I said, so every cancer patient that comes through the clinic, whether they're in a trial or not, will get whole genome sequence analysis done. And a lot of the problems that you're talking about are actually being addressed by the big sequencing companies. So, there are 3 or 4 major players like Illumina and Life Technologies. And they're all addressing those issues. So there are huge international consortia coming together, linking, for example, within the US, we're one of 9 clinical centres who are all linked into the same project. And they're addressing those problems, so they're providing all this really detailed bioinformatic work that's required, bioinformatic support. They're using cloud computing for storage of data and all that sort of thing. So that's been taken care of. There's no illusions that there's a long way to go, but I think it's really taking a lot of attention. There's a lot of investment, and it's coming together. And in a lot of situations, particularly in the clinical trials that we're doing, where you're looking at specific markers within specific genes, so you pull that information out quite quickly. It's the storage of the whole genome and sequences, and then retrospectively going back in to analyse that data that's going to be more complex. One thing that I think people do overlook, though, that there are a couple of really sort of seminal clinical centres in the US who are really using genomic medicine big time. And what they're showing is that use of genomic medicine is not a very simple organisation. There is an organisation which I'm very impressed with in Madison, Wisconsin, who were associated particularly with looking at specific mutations, or trying to find specific mutations that are associated with particular diseases. In order to run their genomic medicine facility, they need 55 staff. And that includes, the technicians, the bioinformatics, it includes the people doing the ethical, all the geneticists. It's a huge, huge effort. It's not something that any centre can just set up an ion torrent machine or whatever, or an Illumina machine, and generate a sequence and think it's all going to happen. It doesn't work like that. You need a massive amount of infrastructure to really deliver that detailed clinical information. But it's coming, in fact, I would strongly recommend, there's a genomic medicine conference that happens in March in San Diego, which I've been for the last 2 years, and you just see huge, huge changes at every meeting you go to of how things are advancing. And I think there's a lot, what they're doing in the US lends a lot of credibility to the whole evolution of genomic medicine, and there are a lot of lessons to be learned from going to those meetings and understanding what's happening. But the investment is much greater there too.
Colin, last question.
Yeah, you keep talking about whole genome sequencing, but clearly in oncology you only query a very small genomic space in terms of the druggable genome for oncology drugs.
Well, you can sorry, you're right.
And the actual technically, is it actual targeted, large-scale targeted resequencing you're doing?
No.
Rather than exome sequencing?
No.
Genome sequencing.
There are 2 elements to that. So, in fact, you'll find that Life Technologies have a cancer exome product, which is a specific next-generation sequencing product, which is looking at about 50 genes, I think, 50 specific genes. It's somewhat related to the thing that TSB and CRUK were running, where I think they're looking at 14 particular targets. So there is a commercial product in the US, cancer exome sequencing, which is looking at about 40 different genes. There's also, as I said, there is the whole genome sequence analysis as well, because nobody really knows in the future what's going to be done. And a lot of pharmaceutical companies just want to basically store down the data so that they can do retrospective analysis, and they're not presupposing which genes they're going to be looking at in the future. And hopefully as the bioinformatics comes along, so you can look at the mutational patterns that are associated with response to individual drugs in cancer or anything else, then you can look at that, you can look at clinical outcomes for the individual patients. So there are different levels to the analysis that are being done for specific requirements.
Okay, thank you.
Thank you very much. Okay, so I'll close this session. I'd like to thank